Hi, I'm Emma, and I'm going to be talking about inheritance patterns. Now first, we're going to be talking about Mendel's genetics. First, I'm going to be talking about why the extensions of Mendel's genetics are very important. Well, to begin with, they can be used to detect disorders and diseases before people are diagnosed, and sometimes even before birth. Second, we know a lot more about how diseases can be inherited, which is very important. If you still don't see why it's important, I'll just give you an example with blood type. When you're receiving blood, if you don't receive the same type of blood as you have, then you could easily die or run into major issues. Without the extension of Mendel's genetics and knowing about it, we wouldn't be able to know things like this. Now for inheritance patterns. Starting out with polygenic inheritance, one characteristic can be controlled by two or more genes. If you break down the word polygenic, the first part poly means many and the second part genic means genes, so it makes sense as to why characteristics can be influenced by multiple genes. An example of this in humans would be height, skin color, and eye color because they are all influenced by more than one gene. Next, we have multiple alleles. For this, 3 or more forms of a gene can occupy the same place, but only 2 alleles can be present. An example of this would be blood type, where as you can see, there are 4 different blood types. And it's called multiple alleles because there's obviously multiple alleles. As you can see, there's a big IA allele, big IB allele, and then the little I allele. And with inheritance patterns, these two are dominant, while the little I is recessive. And how that works is if you receive from both parents the big IA allele, or you receive one big IA allele, one little I allele, you will have blood type A. Now, if you receive big IB from both parents, or big IB little I, then you'll have blood type B. And to get blood type O, you obviously have to have both recessive or both of the little I's, then you'll have blood type O. And blood type AB would be if you receive big IA and big IB, and then you have blood type AB. Now for codominance, when a gene is heterozygous, both alleles can be expressed. Now an example of this would again be blood type, but only if you get AB blood, where you inherit type A from one parent and type B from the other parent to get type AB. Now this may make more sense if you use markers to represent the blood type on the red blood cells. So for blood type A, let's say the markers look like this. Now for blood type B, let's say the markers look like this. Now if you have blood type AB, then your blood cell would look like this. As you can see, codominance, both alleles are expressed. As you can see, this blood cell has both of the markers, A and B. If codominance is still confusing, think about a Punnett square where two parents with AB blood are being crossed. There's a 50% chance of getting AB blood, a 25% chance of getting type B blood, and a 25% chance of getting type A blood. Now both the A and B alleles will take dominance in type AB blood, whereas only one of them take dominance here. At type AB blood, both of them have dominance, and therefore it's codominance. Incomplete dominance can often be confused with codominance. The difference is that in incomplete dominance, one allele isn't fully expressed over the other allele, and they kind of mix together in a way. For clarification, we can make a Punnett square and use an examples crossing red flowers and white flowers. For a cross between a red flower and a white flower, as you can see, the results will have one red allele, one white allele. Now neither of them take dominance because since it's incomplete dominance, they're gonna mix together like I said before. So the result will be pink flowers because red and white makes pink. So when the alleles mix, it'll make a pink flower. And if you were to mix a red flower with a pink flower, you would get 50% of the offspring being red flowers and 50% being pink flowers. Another part of inheritance patterns is epistasis. This is where one gene can mask the phenotype of another gene. Now let's say you have 4 flowers which are determined by the B alleles. The big B is dominant over the little b, and the big B is going to make the flower white. Now, if you are homozygous recessive and have little b, then your flowers can be blue. Now, this is going to change once the E allele comes in. If the E allele is dominant, homozygous or heterozygous, then the flower color will stay the same. But if it's recessive, like it is here, the flower color will change. So let's say the E allele makes a flower black. So this flower would now be black, and this flower would now be black. And it doesn't matter what the B allele says, because as long as the recessive E allele is there, the flower is going to be black. Well, I hope that all made sense and that you now have a better understanding of inheritance patterns and the importance of the extension of Mendel's genetics.